Now this is our modern model of the circadian molecular clock. It might seem kind of intimidating to look at it all in one piece right here, but we're going to go through it one step at a time. So let's get rid of the vast majority of this stuff and consider what we've already discussed so far. We have clock and cycle, these black ovals that are on the promoter region of a number of different genes. Clock and cycle form a heterodimer. They bind together, which allows them to bind to E-box, which is just a specific promoter. E-box is a promoter that's found on a number of different genes, not only period and timeless, but many other different genes. So clock and cycle are activating not just the period and timeless genes, but many, many others. And so they control lots of different genes that have to do with circadian rhythm and circadian outputs. Now, before we move further, we should make note here that the dotted line here represents the nucleus, which is labeled. Outside of our dotted lined oval, we have the cytoplasm, right? So we're still inside of a cell, but the cytoplasm being the outside of the nucleus within the cell, okay? So clock and cycle are transcribing genes in a circadian rhythm. In this little sine wave, this sine wave symbol here with night under it shows that the peak of transcription is happening at night, which is what we saw in timeless and period. Peak mRNA is early subjective night. But we're not too concerned about the output of these like sort of peripheral clocks, peripheral genes. We want to know specifically the mechanism that makes this circadian. So let's go over just these in-house, these transcription-translation feedback loops that control this cycle, this clock and cycle transcription. So in addition to making these other clock genes, we've already established that it also transcribes per. So we get a circadian rhythm in a clock and cycle are producing per RNA, which leads to the production of per protein in the cytoplasm. Now, one of the problems with the initial production of per here is that there is another protein, a DBT, which is short for double time. And what this does is it rapidly degrades per in the cytoplasm. And per production is ramped up relatively quickly. Lots are made, and right away, lots of it is degraded. Clock and cycle are also producing the timeless mRNA, which produces the time protein. Now, what's special about this is that time saves per from degradation. There are two proteins, SGG, which is short for shaggy, and CK2, casein kinase 2. And what they do is they phosphorylate the per and tim dimer. And when per and time form a dimer, it actually forms a dimer with double time. So it's actually three proteins that are bounded together. And per and tim are phosphorylated by shaggy and casein kinase 2. So what's the significance of the phosphorylation? Well, the phosphorylation allows this protein complex to enter the nucleus again. Without it, this complex would be stuck in the cytoplasm and couldn't gain entry to the nucleus. Once in the nucleus, this protein, this triple complex, double time period and timeless, acts as a repressor on clock and cycle. Clock and cycle no longer are able to facilitate the transcription or activate the transcription of per and tim. And this is the primary transcription-translation feedback loop of the circadian clock. However, it's worth going over some of the other details in the secondary and tertiary clock. So another cool detail about this primary TTFL, transcription-translation feedback loop, is the production of another protein that's under the influence of the master circadian transcription via clock and cycle is cryptochrome. CRY, the cryptochrome protein, is produced like the other proteins in the cytoplasm and builds in abundance along with the proteins. And what it does is it has the ability to kind of do what double time can do to period. Cryptochrome degrades timeless. But a very interesting thing here, it only has this effect when it does this in the cytoplasm and in the nucleus. So cryptochrome is found in both. It can easily get back into the nucleus. So it can degrade timeless in both places. But it only does this when it's activated via blue light. All right, so the organism, this timeless breakdown only happens when the organism is exposed to light. So this timeless breakdown only happens during the day. And so, or the exposure to artificial light. Right? And so this is part of the primary feedback loop. Now let's talk about the major secondary feedback loop. It involves the production of one half of our activator, our E-box activator, and that's clock. So clock has its own gene from when it's produced, and its production is also has a circadian transcription-translation rhythm to it, and it's produced via activation of the VP box. So not the E-box. So clock and cycle does not turn on the clock gene. It's produced in the cytoplasm. And of course, we know that this feeds back into the nucleus, and it takes part in activating E-box, which is our primary feedback loop. Now, E-box is also activating a few other proteins that are involved in the secondary loop. VRI is produced, and so is PDP1, which stands for par domain protein 1. These two proteins are being influenced. So what is their role in our secondary cycle? They both feed back onto the VP box. Now, VR1 is fast. It's produced fast, kind of like how period is, and it has an inhibitory effect on reducing, of inhibiting the production of the ultimate production of clock. PDP1 is a slower loop, and it comes in after VR1 and initiates it activates the transcription of the clock mRNA, the clock gene, which gives rise to our clock protein. And this cycles in such a way in which the inhibition of clock via VR1 is occurring at the same time in which our double time period timeless complex is inhibiting clock and cycle on EBOX. So it reinforces this negative arm of our circadian molecular cycle. And this is why it's secondary. It's supporting the primary, the negative feedback arm of the primary cycle. And this is our secondary transcription-translation feedback loop. And there's one more small feedback loop that I want to mention here as well. And this is the production of Clockwork Orange. And so this is under circadian control via the EBOX activation. The CWO, Clockwork Orange mRNA, is produced, which produces Clockwork Orange protein, which feeds back onto the EBOX itself, repressing, inhibiting that production. So it's sort of a redundant mechanism that does this at the same time at which the primary complex is suppressing clock and cycle. So we have multiple layers here, multiple layers of these transcription-translation feedback loops that are affecting this. Now, what I want to do next is show you, sort of on a 24-hour clock, how this is sort of taking place step by step according to a 24-hour clock.